Good evening and welcome. Thank you for joining us for this special event this evening. I'm Dr. David Dodick. I'm the chair of the board of the American Brain Foundation. The American Brain Foundation, as many of you know, funds research across the entire spectrum of brain diseases because many of the biological mechanisms that underlie one disease is similar for other diseases. And therefore, we believe funding research across a wide spectrum of them is our best hope, really, for finding improved treatments, preventions, and ultimately cures. I truly believe that a seminal discovery in one area of brain disease research will have a profound ripple effect on our understanding and treatments of others. So that's why I'm excited for the opportunity to speak with and introduce you to some of the American Brain Foundation's next generation research grant recipients who are working to find the brain disease cures of tomorrow. And joining us tonight are members of the 2022 class to share more about their innovative and cutting edge research projects. And they truly are innovative. I would never have been able to compete with them. So I'm glad they weren't around 30 years ago. After they share, we will transition to a question and answer where you can ask our researchers any questions you may have. And throughout the webinar, please feel free to put your questions in the chat, and we will get to as many, I promise you, as we can during the question and answer. And so I hope tonight you come away with the hope that a future without brain disease is possible because of research projects like the ones you'll be hearing about tonight. But first, let me just tell you about the American Brain Foundation's Next Generation Research Grants program. It's through these grants we fund and support really the best and the brightest early career clinician scientists doing research that promises to yield results that I think will directly impact patients. And over the years, the program has provided a total of $34 million to almost 300 researchers, the vast majority of whom have gone on to secure additional funding from the NIH and from other national funders and have gone on to illustrious careers in research. So we want to thank our funding partners for making this program possible. And there are a lot of them, and we're very grateful for that. And I want to give out a special thanks to our founder and our research partner, the American Academy of Neurology. Supporting early career research is really important. I was one of them I was just telling the group here, I was one of them about 30 years ago. And it really was the thing that really kickstarted my career. Several of our board members of the American Brain Foundation, several department chairs across the country have all been recipients of these early career research awards and have gone on to make significant impacts on neurological disease. So these are extremely important and something we're very proud of at the American Brain Foundation. So this program really truly exemplifies our mission of bringing donors and researchers together so that one day we can all live a life without brain disease. So without further ado, let's meet our researchers and hear about their work. So first, we're going to start with Dr. Kleiman from the University of Miami School of Medicine, and he'll be sharing his work around cognitive aging and age-related memory loss. Dr. Kleiman, can you tell us about your project, please?
Sure. Thank you, Dr. Dodick. I'm Michael J. Kleiman. I'm a postdoctoral research fellow at the University of Miami Miller School of Medicine. And as a recipient of the McKnight Research Scholarship in Cognitive Aging, I'm seeking to develop new methods to measure the very subtle ways our brains change as we age. Aging is a part of our lives. There's no avoiding it. Just as our bodies age along with us, so do our brains. Our cognitive processes slow down ever so gradually as we grow older. However, because these changes are so gradual, there aren't many great ways to measure exactly how these changes progress over time. My approach is to measure speech and language using new advances in artificial intelligence technology, so we can paint a better picture of what normal cognitive aging looks like. First, I ask participants to perform a handful of very simple tasks, such as describing everything happening in a picture. Then, using the same technology found in Siri and Alexa, I take the recorded audio and create a transcript. And from this transcript, I can measure a huge number of things, like the complexity of the word choices, how detailed the descriptions are, and how often pauses like or are used. I also use the audio recording itself, which can give me information on physical characteristics, such as wavering and pitch. From all of this, I analyze the data to determine what subtle aging looks like. For example, while language complexity probably wouldn't be affected much with age, we might find that the length of pauses before those complex words or phrases might increase, as it might take just a little bit more time to find the right words. I also look for comparisons to the participants' demographics, like sex and race, or other diseases they may have, so we can create a more detailed model. Of course, there are other benefits to this research than just understanding how we age. In order for us to better identify abnormal decline, such as dementia in its earliest stages, we need to understand what normal decline looks like. My hope is that from this research, we are able to better map the progression of normal decline, allowing us to notice the early signs of Alzheimer's disease and other dementias earlier than currently possible.
Dr. Kleiman, thank you. Really, really important work, especially because we all know that Alzheimer's disease begins in the brain sometimes 15 to 20 years before the onset of symptoms. And so these subtle nuances that change without anybody really noticing, including the patient, is critically important. So thank you for that, and look forward to asking you some questions about that. If anybody has any questions regarding speech and cognitive aging and Alzheimer's disease, Dr. Kleiman is your man tonight. Okay, on to Dr. Sikorsky from George Washington University, whose research involves understanding specific autoimmune factors related to myasthenia gravis. So Dr. Sikorsky, please.
Sure, thank you. Thank you so much for your introduction. It's really a pleasure to be here to talk to you about my research. So my name is Patricia Sikorsky, and I'm a postdoctoral research scientist at the George Washington University in Washington, DC. So the focus of my research is a rare neurological autoimmune disease called myasthenia gravis, or MG. And specifically, I'm interested in the B cells in these MG patients. So a B cell is a specialized white blood cell that makes antibodies to prevent bacteria or viruses from infecting us and making us sick. But occasionally, these B cells can make antibodies that can attack our own bodies. So in the case of MG, it's caused by antibodies that disrupt the communication between the muscle and the nerve, causing muscle weakness in these patients. There is no cure for MG, and the current treatments can improve symptoms of muscle weakness, but the improvements in these symptoms can vary quite significantly, depending on which type of antibody that patient has. So there's really a great need for more specific treatments for MG. So about 80% of patients have antibodies against a protein called the acetylcholine receptor, and 5 to 8% have antibodies against another protein called muscle-specific kinase, or MuSK. So the B cells that make these antibodies in acetylcholine receptor positive and MuSK positive MG make different types of antibodies, or we call them subclasses. And what that means is they have different properties and functions that can disrupt that communication between the muscle and nerve in different ways. So these differences contribute to some of these differences in the treatment response for these patients, but scientists don't really understand why or what the events are that cause the B cells to make, from these MG patients, to make different types of antibodies. If we understood this process, we can definitely find ways to therapeutically target these B cells to prevent them from making those types of antibodies. So this award will allow me to really investigate the inner workings of these B cells from MG patients to discover how they make these different types of antibodies. And my preliminary findings indicate that the types of genes or pieces of DNA that are activated in the B cells from patients with acetylcholine receptor positive MG differ from those in the B cells from the MuSK patients. And the genes that I've identified so far are part of a network of signals that could influence the type of antibodies the B cells make. So I'm really excited to continue working on this project, and I'm deeply grateful for the funding provided by the Myasthenia Gravis Foundation of America, the American Brain Foundation, and the American Academy of Neurology, which will allow me to continue this research. So thank you.
Thank you, Dr. Sikorsky. Terrific work. Look forward to seeing the results of your work. Our next speaker is and researcher is Dr. Mackey, who comes to us from the University of Colorado, Anschutz Medical Campus. So Dr. Mackey, can you tell us about your research examining aggression towards caregivers in Lewy body disorders?
Yeah, thank you for that introduction. So just to sort of preface this work really stems from my research done during my research fellowship, actually, which just finished, in which sort of the jumping off point for this was recognizing that aggression is probably a more prevalent issue than we had previously understood in Parkinson's and Parkinsonian conditions. In two separate clinical trials, we observed an incidence and prevalence of near 20% amongst caregivers who were reporting aggression from patients. And that was sort of served as the jumping off point for diving into this issue a bit deeper. And so out of my research fellowship, I was able to show that it wasn't simply cognitive impairment, medications, or a lot of the other factors that we assumed would contribute to patients exhibiting aggressive behavior, things like dementia. But rather, it was much more complex than that and related to issues pertaining to caregiver-patient relationships, grief and coping with illness, and significant loss associated with their disease, as well as some aspects of cognitive impairment, some of those things that we expected. And so my ultimate goal is to understand ways that as neurologists and movement disorders specialists, we can support caregivers and engage with caregivers who are really the ones bearing the brunt of these types of behavioral changes. And so the work that I'm planning to do with the funding through the American Brain Foundation and the support from the American Academy of Neurology is actually look at what are our current practice approaches amongst movement disorder specialists for addressing this issue in particular. Because in order to move the needle on engaging specialists and supporting caregivers, we need to understand what currently is being done to address this issue. And less to say, this is something that had not really been studied in any particular way in Lewy body disorders. And so, I think the funding and support that I'm being provided through this to really dive into this deeper and ultimately come out with some ways to help our caregivers and in turn help support our patients through this disease.
Thank you, Dr. Mackey. Very clear. Our next speaker is Dr. Shaiko from the University of Calgary, Alberta Children's Hospital, and her research is looking into sex differences in Tourette's syndrome. So Dr. Shaiko, please tell us more.
Thank you so much. I would like to, again, thank you, American Brain Foundation and Tourette Association of America for giving me the opportunity to conduct this research. So I'm a neurologist and I'm a movement disorders fellow in the University of Calgary. Well, I'm for many years already, I'm dedicated researcher and also clinician focused on patients with Tourette syndrome. Probably you have heard that Tourette syndrome is neurodevelopmental disorder that is characterized by tics, motor and vocal tics, with the onset prior to 18 years old. And well, up to this date, it's actually not really clear what is the trigger for this disease. There are probably different triggers. Actually, there is not much when it comes to sex differences in research dedicated to this topic. What we know is that it's more prevalent among males, and that from our preliminary data, it seems that in females, it has more severe course, for example, when it comes to some particular types of tics. But the main goal of our study, first of all, is to explore clinical phenomenology of tics and comorbid psychiatric conditions, because in 80–90% of patients, psychiatric comorbidities exist. So we want to compare males and females in cohort of our patients in Calgary. And also, we would like to explore whether there is any influence of sex hormones and menstrual cycle in females on tic severity. And finally, we will also conduct a neuroimaging study comparing males and females. And we hope that with this, we will also, well, first of all, we will help our patients, and we will also find some differences when it comes to treatment for males and females. Well, I hope that at the end of this 2 years, I will be able to give you some answers. Thank you so much.
Thank you, Dr. Shaiko. Excellent work. The next researcher we'll hear from tonight is Dr. Lemley from Brigham and Women's Hospital in Boston, and her research aims to explore the potential connections between the gut microbiome and drug-resistant epilepsy. It seems that the gut microbiome these days connects to almost every neurological disorder. So it's a pretty exciting area of research. So Dr. Lemley, looking forward to hearing from you.
Yes, thank you for that introduction. So it is very exciting. I think there's been a tremendous amount of papers written here in the last and published in the last few years. It's really an exciting area. So just for some background, epilepsy is an incredibly common disease, and I'm an epilepsy doctor. So 1 in 26 people have epilepsy. Most of our patients, fortunately, do really well on the first couple of medications that they try, and their seizures can be completely controlled on 1 or 2 medications. But a lot of people aren't so lucky. So we think 1 in 3 people will have what we call refractory epilepsy, meaning that their seizures are not controlled on medications. And there's been quite a few new medications that have come around in the last 10 years to help control seizures. But unfortunately, they just haven't had a big impact on these particular patients so far. And so we have to think about why medications are failing, what else is going on. Medications may fail for many reasons. Maybe a medication is causing too many negative side effects. Maybe a medication is not getting to where it's supposed to be. So it's just not getting into that brain tissue that's causing seizures. Some other good questions to ask are, how do we make our current medications more effective? How do we make them more tolerable? Or are there other mechanisms completely that we should be thinking about? So I believe that the answers to these questions might be in our guts. And so yes, I'm talking about our intestines. And in particular, I'm talking about the bacteria that live within our intestines. It's also referred to as the microbiome. So our gut bacteria can do many, many incredible things. They can make some of our medications inactive or not effective. They can also reactivate some of our medications, so making them effective once again. They produce many important brain chemicals. So they can produce serotonin. They can also produce very important neurotransmitters or chemicals that act in the brain, such as GABA, which is a very important chemical in epilepsy. GABA can stop or inhibit seizures. And there's been some very exciting previous research in the last 10 years or so that has shown how, in mice at least, if you change the gut microbiome, you can actually change how likely it is for that mouse to develop epilepsy. And in human studies, and it has been shown that people who have this difficult to treat epilepsy, their array of gut bacteria are different compared to those who have epilepsy that's easier to control. So my focus, my research focus will really be on trying to figure out what bacteria alter our anti-seizure medications. And I'm going to be doing this by taking stool samples and analyzing it from patients who have epilepsy. So I'll be sequencing that bacteria out and figuring out what bacteria are in which patients. And I'll also be identifying what I'm referring to as good bacteria and bad bacteria in epilepsy. Meaning I'm going to figure out which bacteria make anti-seizure chemicals and which bacteria make pro-seizure chemicals. And that's going to be done by analyzing chemicals in blood samples in patients with epilepsy. So my overall goal is to understand how we might be able to manipulate the gut bacteria to make our drugs either more effective, more tolerable, or create a more anti-seizure environment within someone's body. So I'm very thankful to the American Brain Foundation for supporting my research. I think that microbiome work in any neurologic disease really represents the goal to cure one, cure many. For example, just in some other diseases, we're learning more and more about how our gut microbiome interacts with our immune system. So there's some exciting things happening in the realm of multiple sclerosis with that. We've also actually learned that there is one bacteria that alters the main drug that's used to treat Parkinson's disease. So the presence or absence of that bacteria can really change how well that medication is working. So I think by exploring the microbiome in epilepsy, we're really going to get to understand a lot of other neurologic diseases. So again, thank you very much. It's really exciting work.
Thank you, Dr. Lemley. Fascinating. And thank you for tying this cure one, cure many concept together for us and for our audience here tonight. Speaking of that, our next speaker and researcher is Dr. Motter from Columbia University. And he's looking at olfactory functioning across dementia diagnoses. So across the spectrum of dementias, what is the role of assessing for olfactory function, and what is the mechanism, I suppose, of that loss, and what are the implications for these diseases? So Dr. Motter, looking forward to hearing you.
Thank you for the warm introduction. It's a pleasure to be here with you all tonight. My project comes from a desire to reduce the length of time and uncertainty that happens early in the diagnostic workup phase of dementia. It sometimes takes a long while to arrive at a diagnosis, and there's often a risk of misdiagnosis. Smellability provides clinically meaningful information because loss of smell predicts increased risk of developing dementia. Smell problems are seen in many types of dementia, including Alzheimer's disease, frontotemporal dementia, dementia with Lewy bodies, and vascular dementia. Loss of smell is useful for helping distinguish these dementias from healthy aging, though it's unclear if different types of smell abilities can also tell these dementias apart from one another. And we don't really know much about whether smell ability is worse in some dementias than others. There are several smell abilities that I'm examining in people with dementia. One is odor identification. If you sniff your cup, can you tell if it contains coffee? Another one is odor threshold. How well are you able to detect very faint smells? And the last is odor memory. If we take away your coffee cup and bring back several drinks later, can you remember which that you've smelled before? Finding unique patterns of these smell abilities that are specific to each type of dementia may help us gain diagnostic clarity during evaluations. Some smell abilities might be badly affected in some dementia types, and other smell abilities may still work very well in other dementia types. So to address this for my project, I'm recruiting patients with Alzheimer's disease, frontotemporal dementia, dementia with Lewy bodies, vascular dementia, and older adults without dementia. They're completing these smell tests and tests of thinking abilities. Some types of patterns that I think might emerge will be that Alzheimer's disease patients will have worse odor memory than the rest, whereas dementia with Lewy bodies will have worse odor threshold and odor identification than the others. And all dementia etiologies, except for vascular dementia, may have a pronounced overall smell profile that's worse than otherwise healthy aging. And although this project is trying to find unique aspects of each dementia, it's also trying to create a group of tests that can be used to help the diagnosis of any dementia, kind of in line with the cure one, cure all. If we can better diagnose dementia in the early phase, we'll be sorting people into the right treatment trials for later drug and intervention discovery. And this project only exists through the generosity of the American Brain Foundation, the American Academy of Neurology, and the Alzheimer's Association. Thank you all very much for your support.
Thank you, Dr. Motter. Fantastic. You know, it was, you really tied that together so nicely to the cure one, cure many approach. And, you know, we started off today by talking about why the American Brain Foundation funds research across the spectrum of neurological disorders and how the mechanisms that underlie one are often very similar to the mechanisms that underlie another one. And your research project is a beautiful example of that. So thank you. Next up is Dr. Arnold from the University of California, Irvine, who's looking at new genetic risk factors for amyotrophic lateral sclerosis. Dr. Arnold.
Hi, well, thank you for this opportunity to tell you about my research. I'm a postdoc in the Laspada lab at UC Irvine, and this project, which was funded by ABF, is building upon exciting preliminary data which revealed that an understudied aspect of gene regulation called alternative polyadenylation both underlies a novel genetic risk factor for ALS and occurs broadly in neurons affected by ALS. And so in collaboration with another group at UC Irvine, we applied a new computational tool to data sets that contain the genetic code of thousands of ALS patients and healthy individuals. And in this way, we identified genetic variants that affect gene function via this process of alternative polyadenylation. These variants do not occur within the protein coding part of the gene. Rather, they occur in a downstream regulatory region known as the 3' untranslated region, or 3' UTR. And we found that mutations that lengthen the 3' UTR of the gene ataxin 3 are associated with risk of developing ALS. And importantly, there are existing gene therapy tools to modulate the 3' UTR length of a gene. So given the lack of effective treatments for ALS, our goal is to rapidly move this project towards therapy development, both by studying the biological function of the ataxin 3 3' UTR in human motor neurons, and by designing and testing gene therapy approaches, and these would be antisense oligonucleotides, to restore proper regulation of ataxin 3. In parallel, we found that the 3' UTR length of hundreds of genes are altered in neurons in ALS and frontotemporal dementia due to loss or mutation of the protein TDP43. Mutations in TDP43 actually cause inherited forms of ALS and FTD, and TDP43 aggregates are the most common pathological hallmark of ALS. And so we are using computational approaches again to identify the key alternative polyadenylation events that occur in ALS FTD, and we're performing follow-up studies on potential ALS therapy targets.
Fantastic, Dr. Arnold. Really important work, desperate for breakthrough in this area, and it sounds like your work has true translational implications for treatments for patients with ALS. Thank you. Next up is Dr. Szymkiewicz from Vanderbilt University Medical Center, whose research involves looking into potential new treatments for depression in older adults. Dr. Szymkiewicz.
Hi, everybody. Thank you for joining. So my name is Sarah Szymkiewicz. I am an assistant professor and neuropsychologist at Vanderbilt University Medical Center in Nashville, Tennessee. And so my project is titled Non-invasive neuromodulation to enhance targeted cognitive remediation in older adults with depression. And so that's a complex title, but I'm hoping to break it down in these next couple of minutes. And so my project is funded by the McKnight Clinical Translational Research Scholarship in Cognitive Aging and Age-Related Memory Loss through the American Brain Foundation and the American Academy of Neurology. So in older adults, depression is associated with different memory and thinking difficulties. And so the most prominent difficulties are in the area of executive functioning. And so executive functions are higher level thinking skills, like multitasking and problem solving. And so I want you to think of those old cartoons where Bugs Bunny is about to do something naughty and an angel and a devil pops up on his shoulder. I want you to forget about the devil part. And so executive functions act a lot like that angel on your shoulder, and that they help you plan, think ahead, organize, remind you that you're doing X and you need to get back to work, and so on. And so executive deficits in older adults with depression are associated with underlying changes in how the front of the brain functions and how it communicates with other brain regions. And so currently, there are no FDA-approved interventions to treat these cognitive difficulties in older adults with depression. And my project is focused on investigating a few options. And so specifically, I'm going to be looking at a combination of two different things. And so I'm going to look at memory and thinking training, or brain games, with brain stimulation. And the kind of brain stimulation I'm using is a non-invasive form called transcranial direct current stimulation, or TDCS. And so we know that certain types of brain games can improve memory and thinking skills in depressed older adults, but it doesn't work for everyone. And so I'm interested to see if adding brain stimulation compared to a placebo stimulation will augment or enhance the benefit of these brain games. And so this intervention is quite the commitment. It involves coming to Vanderbilt daily for 4 weeks to receive 20 sessions in total. I'm going to be looking at different paper and pencil measures of memory and thinking, as well as brain activity during a task that's done in the MRI scanner to see whether the intervention, which again is the combination of brain games with brain stimulation, changes those things over a 1-month period. And so by doing so, the goal is to improve mood, cognition, and overall functioning in this population to hopefully slow down cognitive aging or reduce the chances of transitioning to dementia. And so this study is a first step towards that larger goal. Thank you.
Excellent, Dr. Shimkovich. Thank you very much. I'm going to come back and ask you later, what is TDCS, and do you actually stick electrodes through people's cranium? So get ready for that. Next researcher here is Dr. Nicolau, who's looking at correction of a common Duchenne muscular dystrophy mutation. Dr. Nicolau, tell us about your research, please.
Thank you for the introduction, Dr. Dodick, and good evening, everybody. It's really a pleasure to be here with you tonight. I'm a research fellow at Nationwide Children's Hospital in Columbus, and I have to start by mentioning how tremendously grateful I am for the support provided by the American Brain Foundation and the Muscular Dystrophy Association, and which is allowing me to pursue this research. As some of you might know, Duchenne muscular dystrophy is a disabling muscle disease that's caused by mutations in the DMD gene. And the most common mutations are deletions of large portions of the gene. Affected boys will typically develop muscle weakness around 2 to 5 years of age, and then usually lose the ability to walk by age 12. And this is followed by weakness of the breathing and then the heart muscles. And unfortunately, life expectancy is usually only in the 20s or 30s. Unfortunately, the treatments that are currently available for Duchenne are rather limited and have only modest efficacy, and in the case of corticosteroids, also many side effects. There are several new and very exciting approaches that are being studied, but many of them rely on producing shortened versions of the dystrophin protein, which are not quite as effective as the full-length dystrophin protein. And this is a major obstacle that's largely dictated by the limits on the packaging capacity that's used, that's available in many of the adeno-associated virus vectors that are used to deliver these therapies. And in combination with that, there are also questions about the long-term durability of some of these approaches. By contrast, gene editing using CRISPR-Cas9 has the potential to put back missing parts of the DMD gene in order to truly restore full-length dystrophin protein expression in a very durable way. And traditionally, this has been done using a system called homology-directed repair, but this is unfortunately a mechanism that's very inefficient in muscle. And for this reason, our approach employs an alternative mechanism called homology-independent targeted integration, or HITI. In order to then deliver the gene editing machinery to the muscle fibers, we package it in a pair of harmless adeno-associated virus vectors that can be injected. And in cells taken from patients with Duchenne muscular dystrophy, as well as in animal models of the disease, we've been able to use this mechanism to replace the missing exons in a range of different DMD gene mutations. And by this process, to restore expression of the true full-length dystrophin protein in the limbs, as well as in breathing and heart muscles. And we're now taking this a step further by looking for ways to enhance the efficacy of the system and to apply it to different types of mutations in the DMD gene. And overall, this is an approach that has the potential to address a very great unmet need by restoring full-length dystrophin expression in patients with deletions in the DMD gene through a one-time intervention. And as my own shout out to the Cure One, Cure Many approach, I have to say that this is also very exciting because HIDI has the potential to be applied not only to Duchenne muscular dystrophy, but also to a range of other neuromuscular and neurogenetic disorders.
Terrific. Thank you, Dr. Nicolau. Very clear, very exciting work. Our final speaker is Dr. Gadani of Johns Hopkins University, who's looking at the question as to why myelin repair does not occur effectively in multiple sclerosis. Dr. Gadani.
Yeah, thank you so much. So yeah, I'm Dr. Gadani. I'm a clinical and research fellow in the Department of Neuroimmunology at Johns Hopkins, and tell you a little bit about my work looking at myelination. So just to summarize, I'm sure many of you are aware, but the brain is composed of neurons, which can be thought of as sort of wires that connect to each other throughout the brain and the peripheral nervous system. And these wires or neurons are insulated by myelin, which is sort of this fatty sheath that's produced by oligodendrocytes, which is another kind of cell. And in multiple sclerosis and several other disorders, it's these cells, or it's this tissue, this myelin, that's actually attacked and destroyed by the immune system. It's sort of an autoimmune attack. And when the neurons don't have myelin, not only are they lacking their insulation so that they can't talk to each other as effectively, but they don't get metabolic support and they get a little bit sick, and they can even die because of the lack of myelin. So basically in MS, myelin is destroyed and ultimately neurons or the nerves themselves end up dying as well. Now, a really interesting thing about our brains is that there's already a mechanism to replenish myelin that's present in all of our brains, and that's called the oligodendrocyte precursor cell. They're prevalent throughout the brain, and in normal circumstances, they're totally capable of regenerating myelin. They can generate new myelin, new oligodendrocytes. But in multiple sclerosis, this does not happen. The oligodendrocyte precursor cells do not make new myelin, and it's really a complete mystery, and it's a really big question in the field about why doesn't this happen, and is there anything we can do about this? So our lab has been thinking about this for quite a while and have identified actually one possible mechanism. And just to summarize briefly, oligodendrocyte precursor cells in multiple sclerosis have been shown to take on a sort of different phenotype or a different sort of role. They see inflammation molecules and become sort of inflamed themselves. And that inflamed oligodendrocyte phenotype is not as good at making new myelin and is vulnerable to killing by immune cells. So basically my project is trying to interrogate that inflamed OPC or oligodendrocyte precursor cell, manipulating it in mouse models primarily, with the objective of one, understanding why doesn't remyelination happen in MS, and two, are there any new targets that could be possible therapeutic targets in the future? And the work is really made possible by the American Brain Foundation. So many thanks to them, and happy to take any questions.
Fantastic. Thank you, Dr. Gadani. Well, I think you can see why we're all pretty excited about the future of brain disease research. You know, we've had 10 here of the 18 recipients of the 2022 CRTSs, and you can also see why we're excited about funding across the spectrum of brain diseases. These are just outstanding projects and really exciting. And so the first question I'll take out of this from the audience is, amazing lineup of panelists. Couldn't agree more. While I'm optimistic to see machine learning and AI applied in brain disease research and diagnostics, having reliable data is probably important to this topic. For example, someone having a speech problem versus someone who is just very nervous that can show similar symptoms. So the question is here for Dr. Kleiman. Is there anything that can be done to ensure confidence in the data and reduce the chance for a misdiagnosis from someone who's just anxious, from someone who's actually developing prodromal Alzheimer's disease or dementia?
I guess that's an excellent question. Well, first of all, it's extremely important to make sure you have a ground truth. If you are researching Alzheimer's disease or dementia or some other disease, you need to make sure that you do your due diligence and get a high quality diagnosis. If you're researching cognitive aging, normal aging like I am, it's really important to do a thorough workup and make sure that there's no other underlying diagnosis and that they do not have cognitive impairment. And so for my project, I'm very fortunate to work with a very gifted neurologist, Dr. James Galvin. At his lab, at our lab, we collect a lot of data on biomarkers for different types of dementia. They have a very thorough cognitive and neuropsychological workup. And we can say that we're very confident in our determinations of whether they are impaired or not impaired.
Dr. Kleiman, what do you think? There are many different areas with regard to speech analytics that you can measure and monitor. What do you think are the most sensitive ones when it comes to dementia or Alzheimer's disease? Or is that part of really what you're trying to answer?
Yes, so obviously if you're going to research normal aging, you also want to keep an eye on how cognitive impairment affects speech. So some of the things that cognitive impairment can impact as far as speech is concerned, hesitations. So people with dementia, with Alzheimer's disease, they will pause, they'll say before short phrases. Whereas someone that does not have cognitive impairment, typically, even though most people will say and normally in everyday speech, it will be before more complex phrases, more complex words. Also, the general complexity of the words, the speech rate also kind of decreases. But it's also important to note that in some studies of normal aging, they've noticed that as age increases, speech rate also decreases, language complexity decreases, hesitations increase. So it's one thing that we're focusing on is we're collecting those biomarkers, those early stage biomarkers for Alzheimer's disease, just to make sure that our cognitively normal healthy controls are actually cognitively normal.
Right.
And they don't have some kind of underlying disease in there.
Right. So maybe a question for Dr. Motter, since Dr. Kleiman's looking at speech as a potentially prodromal indicator of incipient dementia and Alzheimer's disease, you're looking at smellability, olfaction in sort of the same way. But is it true that people will generally notice a loss in their sense of smell? Or is it that only through sort of vigorous testing that you're doing will you'll uncover olfactory loss that isn't obvious to the patient?
That's a great question. We don't see much support for the idea that we are good judges of our own olfactory abilities. There's only a very, very modest relationship between how well people perceive their sense of smell to be and how good it actually is. So it often is one of the symptoms that could develop without people necessarily knowing it. People could have already lost their sense of smell, and because they don't think about it, they don't need to use it as often in their day-to-day life as language, it could certainly be missed. Language is something that would be more noticeable. Word finding problems can be frustrating. They can cause concern. And even if it's due to normal aging, it can be a cause for alarm for someone who's noticed a change. But olfaction, on the other hand, generally, we're not aware of it when we lose it, COVID notwithstanding. And I'm sure a lot of us have noticed a loss of smell from that, though the gradual decline associated with neurodegenerative disorders is usually too slow for us to notice the onset of smell loss.
Yeah, great. A question here for Dr. Arnold. Are individuals with other neurodegenerative diseases more at risk for ALS than those without? You mentioned frontotemporal dementia being associated with ALS in some patients. So are patients with neurodegenerative diseases, dementia, at more risk of ALS?
Right. So I haven't seen any evidence that someone diagnosed with a sporadic form of another neurodegenerative disease, such as Alzheimer's or Parkinson's disease, is more likely to develop ALS. But the interesting thing is that in certain genetic forms of ALS, there's pleiotropy, meaning mutations in the same gene can cause a few different neurodegenerative disorders, but it's more specific. So for example, in a single family where there's the same genetic mutation, the most common one would be a repeat expansion in the C9orf72 gene. You can have frontotemporal dementia and ALS running in the same family. And multi-system proteinopathy is another example where the same mutation in the same gene can cause a different disease in different people. But that's more about the inherited forms of these diseases.
Yeah, thank you, Dr. Arnold. Dr. Shimkovich, I told you I would come back to you. There are a bunch of, well, there are different neuromodulation techniques like transcranial magnetic stimulation that may be effective for depression. You're talking about transcranial direct current stimulation. So can you tell our audience, what is that? And is that something that patients can do at home?
Yes. So TDCS it involves placing 2 electrodes in different spots on the head, depending on what your stimulation areas of interest are. For me, I am looking at stimulation over the front of the brain, and I'm looking at putting stimulation in on the left side, and then it's a circuit, so then it's coming out on the right side. TDCS is essentially broken down. It's an 8-volt battery. It seems on the surface something very kind of scary, but really the strength of the stimulation is an 8-volt battery. And you wouldn't necessarily think that an 8-volt battery is capable of doing anything. But there's some pretty fascinating research out there in the areas of depression and cognitive aging that shows that TDCS by itself or TDCS in combination with a different intervention has some benefits. What they think is going on is that TDCS essentially primes the brain tissue, or it makes the brain tissue more ready to accept an intervention. And so that's why you're usually seeing the most benefit when you're combining it with some kind of with something else. And so, like I said, I'm using it with these brain games. Right now in the United States, trying to think, it is not necessary. I think it's FDA approved, or it's in the process of getting FDA approved for the treatment of depression without any kind of cognitive training with it. It's just being used to improve the mood symptoms. There's several international researchers where their country's equivalent to our FDA has approved TDCS for treatment of depression. And the most recent one that's coming to mind is Korea. If you Google this, there are ways that you can do this at home. I would strongly recommend that you do not do an at-home version and that you do it under the supervision of somebody who is well-trained in these methods, because you can, in those instances, burn yourself where you'd get like a surface burn. But when you do it under the guidance of somebody who's trained in the methods, there are no side effects or no major side effects.
Well,
and there are
Dr.
some approvals for at-home devices.
Thank you. That's wise recommendations. For Dr. Mackey, speaking of brain games, do you think cognitive behavioral therapy will be helpful, could be helpful for those diagnosed with neurodegenerative diseases like dementia?
Yeah, so there's been some research that's looked at this, especially in the Parkinson's world. Cognitive behavioral therapy actually has been shown to be helpful for mood symptoms in patients with those conditions. And it's been studied kind of across other types of dementia-causing disease, Alzheimer's and such. I think, what's come out of that has shown that, especially in mild or early stages of disease, that it can be particularly helpful for patients who are experiencing depression or mood symptoms as just another means for addressing them. Of course, usually it comes coinciding with medications that are used too. But at least there is some indication that for some people that they could benefit from, types of behavioral therapies to help. But of course, it really is limited by the severity of their dementia. If they're unable to, engage with a therapist, it's very unlikely that they're going to get any benefit from that. And this is something we're doing at the University of Colorado, where we actually have a clinic set up for patients with neurodegenerative conditions to work with psychiatrists and therapists who kind of look at their situation, their context in what the sorts of therapies they provide or feel would be useful.
Great, thanks, Dr. Mackey. This is a question for Dr. Lemley. Do you think probiotics or changes in the diet could affect the gut microbiome in a way that would be beneficial for people with drug-resistant epilepsy?
So I love that question because it's such a good question. So I'm going to explain a little bit about the diet thing in particular. So over 100 years ago, some doctors figured out that their patients who have seizures and have epilepsy, if they went into a starvation diet, their seizures got better in some cases. So that was kind of the beginning of what we know of as the ketogenic diet, which is a diet that's very high in fats and proteins, low in carbs. And so for the longest time, I don't think we had a very good understanding on why the ketogenic diet was working in epilepsy. But there has been some fantastic recent microbiome research that's kind of revealed the pathway on why that works. So Dr. Xiao, she's a researcher at UCLA. Her lab published a paper 5 or 6 years ago that showed that it is the changes in the microbiome that is the thing that makes the ketogenic diet worth it, and that's the mechanism. So if you give a mouse a ketogenic diet who does not have any gut bacteria, it won't work. They have to have an intact microbiome, and that you have to see the changes in the microbiome to get an anti-seizure effect. And they can even take the gut bacteria of the mice who have been treated with the diet. They can take the bacteria, give it to mice who aren't on the diet, but give them the bacteria, and they have the anti-seizure effect. So diet and gut microbiome are very much linked, and they really can change seizure outcomes. So that's been well established in mice models. There are similar studies now looking at patients who are with epilepsy who are treated with a ketogenic diet. We're analyzing their microbiomes, some groups are, to figure out what exact bacteria are doing this. So that's very interesting. And there have been some probiotic trials too. Those have been really small studies, and they haven't really narrowed down which bacteria are good and what exactly they're changing. So that's kind of my hope too with some of the studies that I'll be doing, is trying to get more exact and really figure out the exact bacteria that are doing this. Because if we could eventually end up with a probiotic pill with the right combination of things, that could be a potential treatment mechanism in the future.
Great. You're going to be busy for a long time.
I know there's so many avenues to go down.
Dr. Shaiko, you talked about Tourette's in girls and boys. And the question here is, should parents look for different symptoms of Tourette's in girls versus boys? For example, do early symptoms present in similar ways or in different ways in girls versus boys?
Yes, it seems that, for example, some particular types of tics, because we can generally, I already mentioned that tics could be divided in motor and vocal tics, and also another possible sort of division of tics is complex and simple tics. So it seems that in simple tics, they're just, well, just maybe this is something to simplify, is that simple tics are just small movements, and complex tics, well, they just require more groups of muscles. So it seems that in girls generally, at the beginning of the disease is a little bit more mild than in boys. But this changes over time because generally in Tourette syndrome, we know that this disease has very good prognosis. So that after usually the 10 or 12 years, it's getting better spontaneously, even without treatment. But it seems that in girls, this is not always the case. So it seems that although the onset of disease is more mild, then after time it could be that it's not it could be recurring. There could be, and the prognosis is more severe. So there could be, for example, more severe tics later on in life, and also, for example, more co-occurring psychiatric condition. And also it seems that profile of comorbidities, for example, ADHD, is more prevalent in females. So yes, there are differences. Yes.
Yeah. Very good. Thank you, Dr. Shaiko. Well, we're at the top of the hour, unfortunately, and I just want to thank you all for being here today. This is and to our researchers, thank you very much for participating tonight, for sharing your wealth of information, and we're really excited to see incredible discoveries your projects are going to unearth. You're going to make remarkable contributions through these projects, and I know through the projects that you do in the future. So thank you so much for being here tonight. And remember, really, to support more research into brain diseases and disorders, we really need donations to the American Brain Foundation. Our research partners, who you've heard about tonight, provide 2/3 of the funds needed for the programs the research that we fund. And so we rely on the generosity of other donors to make programs like this possible. And you've heard from 10 tonight. We've given 18 this year, but there were orders of magnitude more applications for these awards. So we leave a lot of research on the table, and so we really want to be able to fund the best and the brightest, and as many as possible that we can. We hate to leave good research on the table. So we offer awards every year, and we really need your help to support this next generation of researchers in the future. Every dollar toward research counts, and with your help, we can really make a difference. So I want to thank you all for being here tonight. Thanks again to the researchers, and thanks to the staff at the American Brain Foundation for making tonight possible. Have a good night, everyone.